Yeah. Good afternoon everyone. So here we are in our second session of anatomy of flowering plants, wherein in yesterday's class we have clearly dealt with tissues, complete meristematic tissues, and from permanent tissues we have completed simple permanent tissues. Now we are going to learn and exercise complex permanent tissues. Complex permanent tissues include xylem and phloem. Okay. Complex permanent tissues include xylem and phloem, where if you have to consider advanced division of plant kingdom, that is angiosperms. Angiosperms will have 4 components in xylem tissue and 4 components in phloem tissue. So what are the 4 components of xylem tissue? 4 components of xylem tissue includes, first is tracheids, second will be vessels, xylem fibers, third and fourth will be xylem fibers and xylem parenchyma. Okay. Phloem also has 4 tissues in it, wherein at the center enucleated sieve tube cells will be there. First component is sieve tube cells, and then companion cells, and then you have phloem fibers and phloem parenchyma. Fine, right? So these are 4 components of both xylem and phloem tissues. Now if you see the components of xylem tissue, just we have discussed tracheids, vessels, xylem parenchyma and xylem fibers. Now we will clearly exercise each component of xylem tissue. First is what ma? Tracheids. Remember, tracheids are present in all the vascular plants, that is tracheophytes. All vascular plants are tracheophytes. The name came after the component, that is tracheids. So tracheids are present in all the vascular plants. Tracheids are present in all the vascular plants. All vascular plants include what divisions of plant kingdom ma? Pteridophytes. Take down parallelly. Pteridophytes, gymnosperms and angiosperms. Now let us see the structural description of tracheids. Tracheids are elongated tube-like cells with thick lignified cell walls, wide lumen, and they have tapering ends at the end. They have tapering ends, wide lumen, thick and lignified cell walls. Okay, they are tube-like. One tube is equal to one cell here in tracheids. One tube is one cell. Fine. And they are dead without protoplasm. Walls are lignified, that is sclerenchymatous. Okay, they are dead without protoplasm. Thick walls with variable inner wall thickenings are present. They have thick walls with variable inner wall thickenings, and they are present in all the vascular plants including gymnosperms. Okay. How tracheids are connected to each other? By pits. Okay. Tracheids are connected to each other by pits, and tracheids are connected to other components of the xylem tissue by pits and pit fields. Okay. So they have what? Pits and pit fields. Next, if you see ma, vessels. Vessels are long cylindrical tube-like structures. Vessels are long cylindrical tube-like structures. They are made up of many vessel members. Unlike the tracheids, where one cell is equal to one tube is having one cell, vessel one tube is having many cells. It is made up of many cells called as vessel members. Those cells are called as what? Vessel members. And again their walls are thick and lignified. They have what? Large central cavity. Vessels are having what? Large central cavity. The vessel cell is devoid of protoplasm. There is no protoplasm. Again, vessels are also dead component of the xylem tissue. Vessel members are interconnected through perforation planes. So, how tracheids are connected to other xylem components? By pits. Two vessels are connected through the perforation plates. So, with this perforation plates, the conduction of vessels is maximum when compared to the tracheids. Unlike tracheids which are present in all vascular plants, vessels are present only in gymnosperms. Okay, vessels are present only in angiosperms. Okay, the vessels are not found in pteridophytes and gymnosperms. Vessel members are interconnected through perforations in their common walls. The presence of vessels is characteristic feature only to the angiosperms. Okay, next. Other xylem components, if you see xylem fibers. Xylem fibers, again they are having highly thickened cells. Xylem fibers are again highly thickened cells. They are dead cells with obliterated lumen. Very little lumen is present with obliterated lumen. Very thick walls are present. Obliterated lumen and very thick walls. They may be septate or aseptate. This provides mechanical strength and they are present in all vascular plants again. So, here we have done with three components of xylem tissue. One is what? Tracheids, second is vessels and third is xylem fibers. All the three are dead components of xylem only. Now, the only living component of xylem is xylem parenchyma. Xylem parenchyma is the only living component of the xylem tissue, where this is the only living component and this has thin walled living cells. They are what ma? Thin walled living cells. Their walls are made up of cellulose. Okay, they store the food materials and secondary metabolites like starch, fats, tannins and resins. Ray parenchyma aids in radial conduction of water. Okay, in between two vascular bundles, the cells which are present are what? Parenchymatous. Here xylem parenchyma will conduct what? Radial conduction of water. Next, storage of food and radial conduction of water, and xylem parenchyma is also present in all the vascular plants. Clear, right? The exception is only with vessels. Vessels are present only in angiosperms. Next, if we have to see, the second is what ma? Phloem. Before going to phloem, you should be clear about primary xylem and secondary xylem. Okay. So, primary xylem or secondary xylem it might be. It has all the four components, right? Primary xylem, in primary xylem you have two components based on the pattern of growth. The newly formed xylem of primary xylem is called as protoxylem and the matured form of primary xylem is called as metaxylem. So, how that maturity is taking place, you just see. Okay. Exarch condition, that is in root, where centripetal pattern of growth is observed. Centripetal pattern of xylem maturation is observed. Centripetal pattern. How is protoxylem will be towards the periphery and maturation will be towards the pith. Okay. Protoxylem will be towards the periphery and metaxylem will be towards the center. So, maturation is from periphery to the center. Okay. Maturation is from periphery to the center. Protoxylem, take down ma parallelly. Protoxylem is towards the periphery and metaxylem is towards the center. Fine. This you will observe in roots. Fine. So, this is how the primary xylem is formed in the roots. And in the endark pattern you will see in the stem. Endark pattern in stem you will observe. This is centrifugal pattern of growth. Centrifugal. How is, you see, protoxylem will be towards the pith. Protoxylem is towards the pith and metaxylem is towards the periphery. Protoxylem is towards the pith and metaxylem is towards the periphery. Take down. This is centrifugal. Okay. Protoxylem is towards the pith and metaxylem is towards the periphery. Clear? Understood? Next. So, the same case, same will be the case with phloem also. In roots, phloem, primary phloem also will show exact pattern, and in stem, phloem also will show indirect pattern of maturation. So, this is about primary xylem and primary phloem. Next you see ma, elements of phloem. What are the elements of phloem which are present in advanced division of plant kingdom, that is angiosperms? All the 4 components include, first is what? Sieve tube elements. Sieve tube elements are enucleated. They are enucleated. They doesn't have nucleus. They are enucleated, and it has second component is companion cells, third is phloem parenchyma, and fourth is phloem fibers. This is your diagram wherein you see at the center the sieve tube cells whichever are there, they are enucleated. They can't maintain their own pressure gradient. Okay. At the tips of the sieve tubes you can observe sieve plates are present. Fine. Sieve plates are present on the either ends of the sieve tubes. Next to the sieve tubes we have the, what do we have next to the sieve tubes? Companion cells. Okay. Companion cells are next to what? Sieve tubes. This companion cells will decide the pressure gradient to be floated in the sieve tubes. Okay. So, that is why sieve tubes and companion cells are called as what? Sister cells. Sieve tubes and companion cells are called as sister cells. Both of them are ontogenetically related. Okay. Both of them are ontogenetically related. Next to the companion cells we have phloem parenchyma and phloem fibers. Phloem parenchyma and phloem fibers. Clear, right? So, these are the 4 components that are observed. Let us exercise each component one by one. First it is phloem. It is complex permanent tissue which is composed of what? Sieve tube elements, companion cells, phloem parenchyma and phloem fibers. In gymnosperms specifically, sieve tubes and companion cells are absent. In the place of sieve tubes, they are having what? Sieve cells, and in the place of companion cells, they possess albuminous cells. Okay. Now, let us see the characteristic feature of each element in the phloem. First is sieve tube elements, long tube-like structures which are arranged longitudinally. These are long tube-like structures which are arranged longitudinally. Their end walls have what? Sieve plates. End walls are having sieve plates, and mature cells will lack what? Nucleus. Mature sieve tube cells will lack nucleus. That is why they are called as what? Enucleated. But they contain peripheral cytoplasm and a large vacuole. Sieve tubes will have a peripheral cytoplasm and a large vacuole. Their functions are regulated by what? Companion cells. Their functions are regulated by companion cells. Both sieve tubes and companion cells are ontogenetically related. Take down. Both sieve tubes and companion cells are ontogenetically related, and both of them are called as sister cells. Fine. Next, here you see sieve tubes and companion cells are done. Phloem fibers and phloem parenchyma. Just a second. Yeah, no issues. Here only you see, I'll be explaining you phloem fibers and phloem parenchyma. Phloem fibers, same like that of xylem fibers, they will be aiding in giving mechanical support to the phloem tissue, and they will be having pointed ends, obliterated lumen at the center, obliterated lumen, and with thick lignified cell walls. This is the description of phloem fibers. They are present in all the vascular plants. Okay. Next is phloem fibers. Phloem fibers, they again have thin cellulose. They are made up of parenchymatous tissue. They are keenly involved in storage of again food materials and secondary metabolites. This is about phloem parenchyma. Phloem parenchyma and phloem fibers are clear to you, right? So, with this we have finished out what? Two tissues. What are they? Xylem and phloem, two complex permanent tissues. Fine. Now, we will look into the tissue systems. Tissues are done till here. Now, tissue systems. Look at the tissue systems carefully. Tissue systems, this type of tissue systems based on location and structure. So, keenly remember we have 3 types of tissue system. 3 types of tissue systems. First is epidermal tissue system, second is ground tissue system, and third will be vascular tissue system. Epidermal, ground tissue system, third is vascular tissue system. Epidermal tissue system includes 3 main structures: epidermal cells, epidermal structures like stomata and lenticels, and epidermal appendages like trichomes and root hairs. Fine. Ground tissue system is also called as fundamental tissue system. Fundamental tissue system. Fundamental tissue system or ground tissue system includes all the regions of the plant except epidermis and vascular bundles. Okay, except epidermis and vascular bundles, the ground tissue system or fundamental tissue system includes all the regions of the plant. What does it include, ma? Just take down. After epidermis, hypodermis, right? Hypodermis, general cortex, endodermis, pericycle, pith, medullary race. All this comes under fundamental tissue system or ground tissue system. Clear, right? Next, vascular tissue system. Vascular tissue system just includes the vascular bundles. Vascular bundles. Vascular bundles include what, ma? Xylem and phloem. Fine, right? Take down quickly. And then we will move on to epidermal tissue system of root, stem and leaf. Done, ma? Yeah. Now we will see the epidermal tissue system of root, stem and leaf. Listen carefully. So here, epidermal tissue system. First with root we will see. So root epidermis is also called as piliferous layer or rhizodermis, because the epidermis will be possessing unicellular projections called as root hairs. So these are the epidermal appendages, okay, and there is no cuticle coating over the epidermis of the root. Fine, just 3 points, ma. It is also called as epiblema, piliferous layer and rhizodermis. It is also called as epiblema, piliferous layer and rhizodermis, and because it is having tubular extensions called as root hairs, these are unicellular. There is no cuticle covering over the epidermis of the root. Why there is no cuticle covering? Because the main function of the root is to absorb water and minerals, right? Absorb water and minerals. Cuticle will be having cutin protein that is impervious to the gaseous exchange and water exchange. Okay, when it is impervious to water exchange, how come this will be absorbing the water and minerals, right? So that is the reason cuticle coating is absent in the root epidermis. Second is what is stem epidermis, you see, epidermis of stem. So epidermis of stem will be having cuticle coating over it. Cuticle coating is present above the epidermis. Fine, and they have multicellular hair-like structures called as trichomes. Trichomes are multicellular hair-like appendages, hair-like structures or appendages. Clear, right? So this is about stem epidermis. Fine, ma? Next is leaf epidermis. If you are done with this, I will erase. Okay. Next is leaf epidermis. Leaf epidermis is little different when compared to the root and stem. Yeah. Leaf epidermis. We'll look into both dicot leaf and monocot leaf. Listen carefully, ma. In dicot leaf, you have two surfaces of the leaf. Upper surface is called as adaxial surface or dorsal epidermis. Lower surface is called as abaxial surface or ventral surface. Okay. So the upper surface is called as adaxial or dorsal surface. Okay, and lower side is called, or upper epidermis also you can call it as upper epidermis. Lower side is what, ma? Lower epidermis. And it is also called as abaxial side or ventral surface. Fine. What are the epidermal structures that are present in the leaf? The epidermal structures which are present are stomatal apparatus. They are what? Stomatal apparatus. Stomatal apparatus. Fine. So, not all concepts are related to MSET, ma. Not all concepts are related. So, this anatomy though you can relate it with MSET, okay, and few concepts will be above NEET and MSET level. So, I do not prefer these sessions. You can watch the sessions in the NEET World channel where you will get complete playlist of all the concepts, okay. This is for PGT beta, yeah. So, here what are the epidermal structures you have, ma? Stomatal apparatus. Stomatal apparatus include stomatal pore, and guard cells and subsidiary cells. Fine, stomatal pore, guard cells and subsidiary cells. So here if you see, ma, stomatal pore is at the center, and in the dicot leaf you will have bean shaped guard cells like this, okay, and covering to the bean shaped guard cells you can find the subsidiary cells like this, okay. So more amount of stomatal apparatus you can find in the abaxial epidermis to prevent the process of transpiration, okay. Fine, till here what are the epidermal appendages that you see in the leaf of dicot leaf? You have stomatal apparatus, which includes stomatal pore, guard cells and subsidiary cells. So here if you see, this is stomatal apparatus, right? They are more present in the abaxial surface when compared to the adaxial surface, okay. This is to prevent the process of transpiration. Next, if you see monocot leaf also has two epidermis, upper and lower epidermis, but then here there is equal stomatal distribution, on both the surfaces. Fine. So, stomatal apparatus are equally distributed on both the surfaces. Apart from the stomatal apparatus, there is one more epidermal structure that you can see. Those are called as bulliform cells. What are they, ma? Bulliform cells. Bulliform cells, how can you describe bulliform cells as? Bulliform cells are large colorless cells, okay. They will maintain a shape of monocot leaf both in turgid and flaccid conditions, okay. They will maintain shape of leaf both in turgid and flaccid conditions. Clear, right ma? Any doubt anywhere? No, right? So with this, epidermal tissue system is clear, right? So far what we have dealt from anatomy, tissues are completely done. Meristematic tissues and permanent tissues. What are the meristematic tissues we have dealt with? Primary meristems, secondary meristems. That is based on the origin. Okay. Primary meristems are formed from promeristems and they are involved in the formation of primary plant body. Primary meristems, again based on the location, we have two types. What are they? Shoot apical meristems, root apical meristems, they are apical meristems, and we have intercalary meristems. Intercalary meristems are helpful in regenerating the lost parts of the plant body. Best example is grasses I have given you, right. Next is what? Both apical and intercalary meristems are primarily involved in what? Increasing the length of the plant. After which we have dealt with permanent tissues, wherein all the cells or tissues have gained their structural and functional independent. All the cells are structurally and functionally matured. Fine. So here we have two types of permanent tissues. What are they, ma? Simple permanent tissues and complex permanent tissues. Simple permanent tissues, where all the cells present in the tissue are similar in size. Okay. Complex permanent tissues, where different cells are of different structures performing different functions. Okay. As such, simple permanent tissues examples include parenchyma, collenchyma and sclerenchyma. Okay. Complex permanent tissues include xylem and phloem. In turn, we have learned four components under xylem and phloem. Xylem components include what? Tracheids, vessels, xylem fibers and xylem parenchyma. Okay. Phloem includes four components. What are they? Sieve tubes, companion cells, phloem fibers, phloem parenchyma. Okay. You can refer the book and say. Yeah. Tracheids are present in all the tracheophytes, all the vascular plants. Vessels are present only in angiosperms. Okay. Xylem fibers and xylem parenchyma are also present in all the vascular plants. Next, coming to the phloem. Sieve tubes and companion cells are present in the advanced plants like angiosperms, whereas they are reduced to sieve cells and albuminous cells in gymnosperms. Clear, right? Phloem fibers and phloem parenchyma again you will see in all the vascular plants. Clear till here, right ma? Next we have started tissue systems, wherein we have exercised epidermal tissue system and ground tissue system and vascular tissue system. Epidermal tissue system includes only epidermis. Okay. Epidermis can be single layered, or in few cells it can be multi-layered as well. Epidermis is made up of what tissue? Take down. Epidermis is made up of parenchymatous tissue. Epidermis is made up of parenchymatous tissue. They are long and elongated cells with thin cellulosic cell wall. Fine. They can be unilayered or multi-layered. Multi-layered epidermis you can see in multi-layered epidermis you can see in nerium and ficus. Fine, you can see in nerium and ficus. Fine, right? So this is about epidermis. As such, we have exercised root epidermis, stem epidermis and leaf epidermis. Root epidermis doesn't have any cuticle, they have unicellular projections called as root hairs, and that is the reason root epidermis is also called as piliferous layer, rhizodermis or epiblema. Stem epidermis will have a strong thick cuticle above it, wherein it has multicellular hair like projections called as trichomes. Leaf epidermis, you will have two surfaces of epidermis. Upper surface is also called as adaxial surface or dorsal surface. Lower epidermis is also called as abaxial surface or ventral surface. What are the epidermal structures you can see over here? Stomatal apparatus includes stomatal pore, guard cells and subsidiary cells. More stomata are present in the abaxial surface. And few of the leaves can also have cuticle coating over it. Monocots will have equal to stomatal distribution on the two surfaces. Apart from stomatal appendages, what do you see more ma? Bulliform cells. These bulliform cells are what? Large colorless cells. They will maintain the shape of the cell both in flaccid and turgid conditions. What are these cells as? Bulliform cells. Fine. So, that is all for today's session. In our coming session, we will clearly exercise the charts of ground tissue system, looking into all the regions of root, stem and a leaf, wherein it will be easy for us when we go for the comparative analysis of dicot root, dicot stem and dicot leaf, monocot leaf. And after ground tissue system, we will also deal with vascular tissue system tomorrow. Okay. So, that is all for today's session guys. Thank you.